learners, and welcome to this mini lesson for Chapter 11, Blood and the Language of Hematology. In this video, we're going to cover the components and function of blood and some disorders of red blood cells. So let's get started. If you've taken an anatomy class, you'll remember that blood is actually considered a connective tissue with a liquid matrix. The cells float around in the liquid portion of the matrix. The average adult weighing between 150 and 180 pounds has about 5 to 6 liters of blood in their body. That's the same as about 1.5 gallons or 10 pints. Sometimes we say units of blood. So if we took a test tube full of blood and spun it down in a centrifuge, the heaviest particles would sink to the bottom. These cells are called the formed elements. About 45% of a normal blood specimen is formed elements or cells, and the rest is plasma. And we'll talk about plasma in a minute. So what makes up the formed elements are the red blood cells, the white blood cells, and the platelets. The largest percentage is by far the red blood cells. The term hematocrit, abbreviated HCT, refers to the percentage of total blood volume composed of red blood cells. Normal hematocrit levels for males is 40 to 54%, and for females it's slightly lower, around 38 to 47%. A low hematocrit level may indicate anemia, chronic illness, or blood loss, and a high hematocrit level may indicate dehydration, bone marrow disease, or certain other conditions like pulmonary conditions. So the formed elements make up about 45% of the blood, and the remaining 55% is the plasma. The plasma is mostly water and contains none of the formed elements, but it does contain plasma proteins. It's considered a colloid because it's a liquid with suspended particles. The plasma proteins are albumin, globulin, and fibrinogen. The plasma also transports nutrients, waste products, hormones, and enzymes. If you hear the word serum, serum refers to the plasma without the clotting protein fibrinogen. So if you allow a specimen of blood to clot and remove the clot, what's left is serum, basically plasma without the fibrinogen or any of the blood clotting proteins. Okay, so let's review these terms. RBCs are red blood cells. It's the abbreviation for red blood cells, which is one of the formed elements. And red blood cells are responsible for transporting oxygen. Hematocrit was the percentage of blood volume composed of red blood cells, and HCT is the abbreviation for hematocrit. WBCs is the abbreviation for white blood cells, another one of the formed elements. And white blood cells are active within the immune system. Platelets are responsible for blood clotting, and they are also part of the formed elements. Plasma is a clear yellowish fluid that contains albumin, globulin, and fibrinogen. And serum is identical to plasma, except it does not contain any of the clotting proteins. Now let's talk about the functions of blood. There's 9 main functions of blood, and on this slide I've divided them into 3 sections: transport, regulation, and protection. Transport functions of the blood include delivering oxygen from the lungs to the cells of the body and bringing carbon dioxide from the cells to the lungs for elimination. Transporting hormones from the endocrine glands like the thyroid or adrenal glands to their target cells or organs. Transporting metabolic waste products from cells and organs like the liver and the kidneys for elimination. And transporting nutrients, vitamins, and minerals from organs in the digestive system to organs and cells where they are needed. For example, glucose is stored in the liver as glycogen until we need it in the body as fuel. Regulation functions of the blood include maintaining adequate fluid volume in the circulatory system. This is an act of homeostasis, and this allows the body to maintain adequate cardiac output. This allows for adequate circulation and efficient blood supply to all parts of the body. Maintaining a normal body temperature by absorbing and distributing heat throughout the body when body temperature is low, and encouraging heat loss by bringing blood to the skin's surface. Blood is shunted to vital organs when we are cold, and superficial blood vessels will dilate and bring warm blood to the surface to radiate off heat when we are too hot in an effort to cool the body. Again, that's another example of homeostasis, or a homeostatic mechanism. And maintaining a normal pH in the body. Many blood proteins act as buffers to prevent excessive shifts in blood pH that would alter our normal cellular activity. The body functions at a very narrow pH range, only 7.35 to 7.45, And remember, neutral pH is 7.0. Anything too far outside of that very narrow pH range is life-threatening. On a pH scale, anything below 7 is considered acidic, but in the body, a pH below 7.35 is considered acidic. And the blood does not like to be acidic at all, so the body has a buffer system to maintain the correct pH, and these buffers include things like bicarbonate and phosphate. And finally, the blood also has protective functions. White blood cells, also called leukocytes, along with antibodies and particles called complement proteins that we'll discuss in the next chapter, all work to defend against foreign invaders like viruses and bacteria. And platelets and plasma proteins help form clots which prevent blood loss. This slide asks you to define a whole list of abbreviations, and while it may look a little daunting and you might not remember them all, you will be able to eventually recognize most of them after reading enough lab reports. DL just stands for deciliter or 1/10 of a liter. FL is a femtoliter or 1/quadrillionth of a liter. L is just liter. These are all just measurements that you'll see on lab reports. mcg is microgram, which can also be written with the Greek letter mu, although that can be really confusing, especially if it's handwritten, so we don't use that one a lot. A microgram is 1/millionth of a gram. MCH stands for mean corpuscular hemoglobin. This is the average concentration of hemoglobin in an average red blood cell. MCHC stands for mean corpuscular hemoglobin concentration, which is the average concentration of hemoglobin in a given volume of red blood cells. MCV is the mean corpuscular volume, or the average volume of a red blood cell. PG is picogram, or 1/trillionth of a gram. And TIBC stands for total iron binding capacity, which tells us the amount of iron needed to saturate a red blood cell, which we often measure in anemic patients. The terms on the right also relate to blood. Osmosis refers to the passage of water through a semi-permeable membrane like a cell wall. All cells exchange water by osmosis, including blood cells. And viscosity refers to the thickness of a fluid or how resistant a fluid is to flow. Think of water versus honey. Water will flow much faster than honey because honey is more viscous. Whole blood is about 5 times more viscous than water, although a lot less than honey. So it's true what they say, that blood is thicker than water. Now let's move on to talk about red blood cells. Red blood cells are also called erythrocytes. Erythro means red and cyte means cells. These cells are shaped like biconcave discs, meaning they are flattened discs with depressed centers, and they sort of look like miniature donuts. Mature red blood cells have no nucleus. We say they are anucleate, and basically have no organelles. They are full of a protein called hemoglobin, abbreviated HB, and hemoglobin gives the cells their red color. And hemoglobin is composed of 2 parts, the iron-containing pigment called heme, and that is bound to a protein called globin. The main function of erythrocytes is to carry oxygen from the lungs to the cells all over the body. Interestingly, the cornea are the only cells in the body that do not receive oxygen from red blood cells. In the erythrocytes, oxygen is attached to the hemoglobin to form oxyhemoglobin and then transported to its destination anywhere else in the body. When the red blood cells deliver oxygen to cells, they will pick up the waste product carbon dioxide and then transport that carbon dioxide to the lungs for excretion. Red blood cells will also transport nitric oxide, or NO, which is another respiratory gas that affects blood vessel dilation and smooth muscle contraction. So now let's discuss how erythrocytes are formed and broken down. The process of erythrocyte formation is called erythropoiesis. The suffix poiesis means to make, and this occurs in the red bone marrow. Specialized cells called hematopoietic stem cells become erythroblasts, which are precursors to red blood cells that have a nucleus. These erythroblasts then form into reticulocytes, which are immature blood cells. These reticulocytes are full of hemoglobin and can begin carrying oxygen. They become mature erythrocytes in about 2 to 5 days, and at that point lose their nucleus. You may see a lab test called a reticulocyte count, and this provides a rough index of the rate of red blood cell production. Reticulocyte counts below or above their normal range can indicate abnormal rates of erythrocyte formation. A hormone called erythropoietin, which is released by the kidneys and the liver, controls the rate of red blood cell production. The release of erythropoietin, sometimes called EPO, is triggered by lack of oxygen in the blood. Remember, these blood cells carry oxygen, so if we don't have enough oxygen in the blood, the body gets the signal that we need more, so this hormone is released, and that signals the bone marrow to produce more red blood cells, which in turn will carry more oxygen to the cells that need it. Red blood cell production is also influenced by other factors like iron, B12, and folic acid. The average lifespan of a red blood cell is 120 days. Because red blood cells have no nucleus, they are unable to reproduce, grow, or divide. As the cells age, they become less flexible, more rigid, and more fragile. Small vessels like capillary beds, and especially the circulatory bed of the spleen, trap the small cellular fragments. Macrophages from the liver and the spleen engulf and destroy these dying erythrocytes. The hemoglobin is split into the heme and the globin. The globin is recycled into amino acids. The heme is further broken down into bilirubin, which is a yellowish pigment that is picked up by the blood and sent to the liver and eventually excreted through the feces. The other part of the heme is iron, which is stored by the body for later use as needed to make more hemoglobin. Okay, this slide gives us a quick terminology review. These terms all have the same root, erythro, which means red. An erythroblast, remember, was a precursor to a red blood cell. An erythrocyte is a mature red blood cell. Erythropoiesis is the process or formation of red blood cells. And erythropoietin is the hormone secreted by the kidneys and liver that stimulates red blood cell production. Now let's talk about disorders of red blood cells. Anemia is the most common disorder. Anemia is an umbrella term, and it simply means the blood's oxygen carrying capacity is too low, so there's not enough erythrocytes or not enough hemoglobin. Symptoms of anemia include shortness of breath, which is abbreviated SOB, fatigue, paleness, or sometimes called pallor, and feelings of being cold or chilled most of the time. Under a microscope, the erythrocytes may appear hypochromic or pale due to the lack of the red-colored oxyhemoglobin. In the term hypochromic, hypo means below or deficient, and chrome means color. There are several types of anemia, but there are three main categories. Blood loss, not enough blood cells being produced, and too many blood cells being destroyed. Your book doesn't mention the first category, blood loss. This is hemorrhagic anemia, and it can be acute or chronic. Acute blood loss, like from a stab wound, can cause a severe deficiency, but it can also be caused by chronic blood loss from an undiagnosed bleeding ulcer or heavy menstrual bleeding. Once the cause has been identified and treated, the anemia will usually be resolved. The second category, not enough blood cells being produced, includes iron deficiency anemia, pernicious anemia, aplastic anemia, and one your book doesn't mention, renal anemia. Iron deficient anemia usually results from chronic hemorrhagic anemia, like from an ulcer or heavy menstrual bleeding, as we said. Remember, the iron from broken down red blood cells is recycled to make new blood cells. If this iron is not adequately replaced in the diet and is continuously lost by the body, the result is a deficiency in iron. Luckily, this can be reversed by treating the cause and taking iron supplements or increasing iron in the diet. Pernicious anemia is thought to be an autoimmune disease that causes the lining of the stomach to stop secreting something called intrinsic factor. Intrinsic factor is necessary for the absorption of vitamin B12. Without B12, the normal development of the erythrocytes is altered, resulting in large, pale macrocytes with limited amount of hemoglobin. Treatment of pernicious anemia includes regular injections of B12. Aplastic anemia results from the destruction of red bone marrow where blood cells are produced. This will affect the production of all blood cells, not just red cells. Aplastic anemia can be the result of certain drugs, chemicals, radiation, or viruses, or the cause can be unknown. Treatment can include blood transfusions and even stem cell transplant. And renal anemia is not mentioned in your book, but I'll touch on it briefly. This is caused by the lack of erythropoietin or EPO, the hormone that stimulates the blood cell production in the bone marrow. Renal anemia frequently occurs in patients with chronic kidney disease because their damaged kidneys cannot produce enough EPO. Fortunately, it's easily treated by administering EPO to these patients. The last category of anemia is when too many blood cells are destroyed. This includes hemolytic anemias. Remember, the root lit means destroy, so hemolysis refers to the destruction of blood cells. There are many things that can cause red blood cells to rupture prematurely, like certain toxins, drug reactions, venoms, transfusion reactions, like if we give someone the wrong blood type, for example. But here we're going to talk about hemoglobin abnormalities, or what we call hemoglobinopathy. In this term, remember, pathy means disease, so we're talking about disease of the hemoglobin. There are two main hemoglobinopathies: sickle cell anemia and thalassemia. Sickle cell anemia is a genetic disorder that's found most commonly in African Americans and Mediterranean populations. It causes the hemoglobin molecule to become deformed into spiky, sharp rods instead of the soft, flexible protein it normally is. When this happens, the red blood cells become crescent-shaped when they unload their oxygen or when oxygen levels are lower than normal, like after exercise or during periods of illness or stress. These abnormally shaped cells are sticky and clumped together, or agglutinate, that means they clump together, and block smaller capillaries. This blocks the blood flow, and the lack of oxygen, or hypoxia, leads to intense pain, referred to as a sickle cell crisis. Sickle cell disease can result in stroke, kidney damage, retinal damage, and heart failure. Thalassemias typically occur in Mediterranean populations. There are several different types, but generally they result in disruption of the globin molecule and leaves the erythrocytes thin, delicate, and lacking hemoglobin. The symptoms can range from mild to so severe that monthly transfusions are required. The last two conditions here are polycythemia vera and poikilocytosis. Let's look at the first one. If you break down polycythemia, emia means blood condition, poly means many, and cyte means cell. So this is literally a condition of an overproduction of blood cells by the bone marrow. Vera is Latin for truth. So in polycythemia vera, the cause is unknown, or true polycythemia. Secondary polycythemia can be caused by lack of oxygen or increased production of EPO, both of which will stimulate the bone marrow to produce blood cells. For example, individuals living at high altitudes often have secondary polycythemia as a secondary response to the low oxygen levels. And finally, poikilocytosis is the medical term we use to describe having abnormally shaped cells in your blood. Poikilo means irregular, cyte means cell, and osis means condition of. These irregular cells could be sickle cells we mentioned earlier, spherocytes, which are dense round cells that don't have the regular depressed centers of red blood cells, or something like elliptocytes, which look like ovals or cigar shapes, or there's several others. But poikilocytosis is just kind of an umbrella term that means you have irregular, abnormally shaped cells in your blood. And finally, this slide gives you some abbreviations from this section of the text. So CO2 is carbon dioxide, that was one of our respiratory gases. HB means hemoglobin. NO is nitric oxide. O2 means oxygen. PA stands for pernicious anemia. And SOB is shortness of breath, or short of breath, depending on the context or how you're writing it. So that's it for this video on blood and red blood cells. In the next video, we're going to look at white blood cells and some disorders of white blood cells. So as always, I thank you for your attention, and I will see you next time.